To be doing is really have a Q&A session. This is a forward-looking statement. And I think, as you all know, we're deep into bladder cancer now. And I think one of the first questions is, why are we different and what's different about our program that compares itself to the current approvals for bladder cancer? And it's a huge market, and BCG, as you all know, is the treatment for patients with bladder cancer. So this is a slide to try and describe very briefly our unique mechanism of action and what may be ultimately in the label, is that the N803, the IL-15, activates the natural killer cell, as well as the T cell, and induces memory T cells. So that's a unique advantage of this molecule, which will take us to the next level. So this is what happens when you give BCG. And BCG induces this stress and activates macrophages. Unfortunately, the results are very temporary. And then, if you then add together with this BCG, this N803, and you'll see this NK cell, this T cell, and the memory T cell activated. And that unique combination of the innate and the adaptive immune system gives you what we call this cell death. And the NK cell, which is unique here, activates and kills and induces this long-term memory. So this mechanism of action of an NK cell, T cell, inducing a memory T cell. So this was probably one of the first of this next generation immune system. We've gone from checkpoints of T cells to now NK cells, and memory T cells will give us a durable, complete remission. What's remarkable is that the results are published now in New England Journal of Medicine of 26 months, the upper end is yet to be reached of this duration of complete remission. So today, we will be revealing at this conference some new data, not just in this BCG unresponsive, but even in the BCG naive population. So this drives a little bit to the mechanism of action of the unique mechanism of action of this molecule, of activating NK cells, T cells, and most importantly, memory T cells. So if you take that concept of a tumor cell, regardless, where a natural killer cell, a killer T cell, and a memory T cell all congregate around the tumor, the possibility of now inducing long-term memory in earlier disease. So what we have under ImmunityBio, which we call ourselves now the Cancer Vaccine Company, I think many, many years ago, 5, 6, 7, maybe even close to 10 years ago, we uttered the word cancer vaccine, and it was considered crazy. Today, as you hear, the concept of a cancer vaccine at today's ASCO is a reality. And we're expanding these options from preventing metastasis to preventing cancer. In order to activate the cancer vaccine, you need more than just the N803, which we show you the bottom portion of that slide, showing NK T cell, memory T cell activation. You also have mechanisms to activate the dendritic cells, and we have a platform of adenovirus, yeast, self-amplifying RNA, and adjuvants. At the end of the day, we can activate your natural killer cells exogenously by giving off-the-shelf a PD-L1 Hank, a CD19 Hank, and soon you'll see this whole concept of a memory-like cytokine-induced NK cell from your own body. And all of that will be modulated with inducers that induce expression, which we call DAMP inducers. That's the platform of ImmunityBio. It's unique in the sense that this is the company that actually has taken on the challenge of bringing all these molecules into play, because there's interplay of these molecules amongst each other. So what we've done over the last maybe 10 years is quietly proven the principle to ourselves. And if you go to clinicaltrials.gov and just type in the word QUILT, Q-U-I-L-T, it's quantitative integrative long-term treatment or lifelong treatment, is we've now taken this to the concept which we knew to prove that not only can we prevent metastasis, can we actually prevent cancer? That's a huge goal. So obviously, we had to start at third line or greater to prove this concept. Then we'd go up to first line, and I'll announce today, we're actually now in trials for patients who are normal. Now that sounds a little crazy, but that's where we are. So let's start here. And obviously, we are known for starting in bladder. And we've gone to BCG, what we call unresponsive, to BCG naive. And we'll talk a little bit about the results. We've gone into BCG unresponsive. We filed the BLA. Unfortunately, we have now a little bit of a delay because there's some questions that's still to be answered on the manufacturing side, which we will address during the question and answer session. We have some very exciting new data on BCG naive, which we will discuss with you today. And then taking from bladder cancer, going to colon cancer, and we'll show you data from a third line colon cancer, and this is the NCT number, in which we have remarkable survival, 11 months, to now moving to microsatellite stable disease, and that's 80% of colon cancer patients that can't be treated today because of the microsatellite stable disease, moving, believe it or not, into normal patients with Lynch syndrome. It's hard to believe that close to 1 in 300 Americans have Lynch syndrome, which states that you have a chance, unfortunate chance, high-risk chance of getting colon cancer and other cancers associated with this genetic disease. And we've just initiated the nation's first trial to prevent cancer in Lynch syndrome. So we've gone from metastatic disease, or advanced disease, that's failed therapy, to prevention of metastasis, to now prevention of cancer. And that's the roadmap which we've taken ourselves on. The question is, does this actually expand into other cancers? And you now hear of the new epitopes and the cancer vaccines that Moderna and companies such as Moderna are developing. And it turns out that we've been in this new epitope and have the only FDA-approved methodology of finding the tumor normal new epitopes, and started this trial very early on in first-line patients, and I'll share with you some of the results today. That takes us then obviously into Lynch syndrome as well. So you're going now from prevention of metastasis to prevention of cancer, and we'll speak a little bit on this new epitope data, which to us is very exciting. We now have 5 patients, and 3 of those 5 patients, the data which I have now, are 4 years without disease. So that first line in pancreatic cancer and breast cancer is the new epitope results, which started in 2019. Then we've gone from pancreas, and we'll show you our second and third line data, moving now into the first line neoadjuvant data. And I think you've seen the data maybe from ASCO today that the neoadjuvant for FOLFOX unfortunately has failed. And we have this plan now with first-line neoadjuvant pancreatic data, similarly with glioblastoma, similarly with small cell lung cancer, and similarly head and neck cancer. So this is the program that we are focusing on at ImmunityBio, called the Cancer Vaccine Company. And we're clearly moving the field from metastatic disease to prevention of metastasis to prevention of cancer. So with that, I'll sort of take questions. Let me see if I can go back to the slide that I think we'll just leave you. Maybe I'll just leave you on this slide so that we can take questions, Kelly.
Thank you, Patrick, for a great introduction. Before we move to cancer vaccine discussion and your unique neoepitope identification approach, we can talk about bladder cancer, because this is one of the major value driver for the company at the moment. So through our interaction with KOL, physicians showed excitement to Anktiva for treating BCG unresponsive population. And you just mentioned you will share some BCG naive development progress. Could you elaborate on that?
Right. So the BCG naive, we've done obviously the phase 1 study, and then now the opening of the Quilt 205, which is the definitive results. What's exciting about the phase 1 study now, it's remarkable, 9 patients. All 9 had complete remission that lasts now 6 years. 2 patients have passed away. That leaves us 7, and we just followed them up now. And they're now 8 years. So what we've now demonstrated for the first time, that by activating the systems of memory, we can actually induce completely long-term memory in these naive patients. So that data we're putting together now, the paper on the first 9 patients has been published, and showing that we can now actually keep these patients completely free of disease for over 6 years, and then those that survive, 7 of the 9, 2 that have passed away not related to cancer, remarkably over 8 years. So this has given us great encouragement with regard to the BCG naive study. And as you know, the numbers of patients is huge. There's close to 80,000 new patients a year. And then when you look at the ongoing data, there's over 500,000 patients that, from on continuing therapy perspective, available just in the United States.
Great, and it's very impressive survival for over 8 years. Curious, have you brought this data set and to the regulatory agency talking about the next step?
Yes, we're in clinical trials, obviously with the BCG naive data, randomized clinical trial, BCG versus BCG plus Na2/3. And these patients that are 8 years in our phase 1 all had BCG plus Na2/3. So that randomized trial is ongoing as we speak today.
Terrific. And also regarding the CRL, I know you cannot disclose details, but we're curious if you could set an expectation on the timeline to move to the next step and solve the CRL.
Right. Well, the good news about the CRL, and we've exposed that at least in our filing, is that it's nothing to do with the clinical data. The FDA has asked us to do nothing more than update the long-term results, which, as you see in the publication, we had 26 months, 24 months, I forget, 26 months. And where we're going now is just to update the longevity, which is yet to be reached. The issues really relate around the contract manufacturing, unfortunately. We used a contract manufacturer to make the drug substance, and they had a big inspection. And we addressing those issues with regard to the inspection this site is making 2 or 3 or 4 commercial products, not just ours. So the inspection was about the entire site. It was a 2-year inspection. And I'm comfortable that we're addressing these issues, and the timeline of from the time the issues are addressed for analysis by the FDA is either 2 months or 6 months.
Terrific. And what is your focus in potential launch preparation? I think you mentioned commercially you will look for a partnership. So what is the status of that partnership talk? And should we expect the partnership exclusively on the Activa commercialization, or any plan for the partnership to go beyond that scope?
Yeah, I think as you see from the slides I just given here on this slide here, clearly the Activa has activity across all tumor types. So we are exploring partnerships because we don't have clearly international, we have US but not international exposure. And that'll be just for the bladder cancer, period. So it'll be separated out from all the other indications. That's ongoing. And, obviously we'll explore that. But we'll get our approval first, and then we'll explore that, I think. I think that's where we're at now.
Okay. How should we think about competition from pembro monotherapy and also pembro BCG combo data, which is supposed to come out soon, as well as other novel therapies in the space, including oncolytic virus and Padcev is also being studied in the same population?
Well, I think 2 things. One, this is mainly a urology disease, meaning the urologists are the treating patients. And they're very not comfortable with checkpoint inhibitors, whether there's handoff. And unfortunately, if you look at the results of the checkpoint inhibitors, there's significant checkpoint inhibitor toxicity. It's associated with checkpoints. We see none of that. In fact, the safety data, which you can see, I think some of it's in the New England Journal of Medicine paper, is remarkable in the sense that it's not unlike BCG alone. So, and then finally, the urologists are very comfortable. There's no change in their practice, just infusing this intravesically, the BCG plus Na+3. So I'm not too concerned about that combination. And I think at the end of the day, what we'll be telling is the results, and the results are, you can do a head-to-head comparison. It's now both public.
Great. And moving to pancreatic cancer, you're running clinical trials in front line, second line, the third line, as in 3 different cohorts in Quilt 99 trial. First, could you elaborate on how's your approach differently from others, and how should we set efficacy bar in each of these settings?
I think this year's ASCO really gives you some insight. This year's ASCO, there's pancreatic cancer in the early setting, in the neoadjuvant setting, and I think that was just been presented yesterday, and I saw the results, and I think the overall survival was 2.6 months or something of that ilk. I think that was done at Harvard, so it's a good school that did the data. Our third line and greater, third line, fourth line, fifth line, and sixth line patients on the left-hand side of the pancreas have over 6 months overall survival. So we clearly are doing something completely different within the world of pancreatic cancer, using, again, this Na+3 activating the NK cell, the T cell. And therefore, we now, with that data, and you'll see that there, the Quilt, I forget the number here, Quilt 88, is now moving into first-line neoadjuvant. And so we slowly will move from these very late-stage diseases, second and third line. What was remarkable, and I made this public, Senator Reid, who had second-line pancreatic cancer, had a complete remission and lived for 2 years. And he's allowed us to publicize that. And to me, that in my career, that was the first complete remission in a second-line pancreatic cancer patient using these techniques. So I think moving to first-line neoadjuvant, which we're in process now of filing that with the FDA as a protocol, is going to be very exciting.
Thank you. Sorry, 88. A better number than 9. And let's move to the exciting cancer vaccine program. Can you first talk about how is your approach differentiated from other cancer vaccine programs, and what kind of validation do you have on your neoantigen identification approach?
That's the approach. And the approach is this unique interaction of a killer cell, a T cell, a natural killer cell, a memory T cell. And, I think if we get that into the label, this will be the first label ever in the history of immunotherapy that has a combination of NK cell, killer T cell, and memory T cell. And that induction just with NA-3 and BCG. When you add this now to the adenovirus or the yeast or the self-amplifying RNA, and as you can see here from the vaccine companies, such as that's presenting right now from the COVID vaccine to the cancer vaccine, that we've been in this field for so long, where even just single agent in our colon cancer model, which I'll go to the next slide here, in that colon cancer model, that third line, I urge you to go look at that NCT number, in which we have 11 months survival, which far exceeds, and that's just the adenovirus CEA MUC1 brachyury. Actually, sorry, that was just adeno-CEA alone in these patients with third-line colon cancer. Now that paper is now published. So I think the evidence, first of all, with the bladder, in which everything has failed, they're unresponsive, they would require cystectomy, getting over 26 months, and yet the median survival is yet to be reached. And in naive, in which you have 8 years of disease-free, and then in colon cancer third line over 11 months, and that's published paper, speaks to the sense that we have found something with regard to the concept of a vaccine. What's exciting now, moving to microsatellite stable, which 80% of colon cancer, which there really is no treatment. We're now moving there, and obviously Lynch syndrome has taken us to patients who are normal. So I think there's some level now of validation of where we're going with this, and then obviously when you add the neoepitope, we're right now in first line. So these patients, 4 patients, 3 patients I just announced for the first time this morning, are 4 years, 4.5 years, free of disease. So I think we have a real shot at creating this vaccine.
Terrific. And lastly, how do you envision the opportunity ahead for your cancer vaccine program along with others?
You mean others in terms of other
cancer vaccine programs?
Oh, how do I rate that?
Big picture.
Okay, well, the big picture is you really have to integrate the NK cell, T cell, memory T cell, all in a integrated fashion. Just CAR T alone, or just checkpoint inhibitor against T cells alone, or just NK cell therapy alone, unfortunately, it does not really work. You really need to integrate that, and this is the only program that's made that decision right from outset, to integrate the adaptive and the NK cell immune system together.
Thank you very much for your time and share your great insights.